Okay, we're going to move into chapter 9, which is lipids and biological membranes. So there's our key concepts that we want to make sure we understand at the end. And let's just start by defining lipids. Now, lipids are the last class of biological molecules that we'll look at, but they are different than the previous ones, the nucleic acids, the amino acids or proteins, and the carbohydrates. These aren't defined by a given structure. They're actually defined by a physical property, their solubility in a non-polar solvent. So there's actually many different kinds of lipids. They serve a variety of functions in and on the body. What they do have generally in common is hydrocarbons or modified hydrocarbon structures. And so how I define lipids is if they ground up a human and they went to extract it into water and gasoline, everything that went into the gasoline layer or the oil layer would be classified as a lipid. So in any discussion of lipids, you typically start off by talking about fatty acids. Now, these aren't really big players by themselves, but they're found as components of other lipids. So you don't have a lot of these free fatty acids, but you do have some, and when they're processed, you do get these free fatty acids. But this is the basis for a big portion of what we'll talk about. So we'll start by talking about fatty acids. And they're classified by their size, the number of carbons that they have, even versus odd. So most natural fatty acids tend to have an even number of carbon atoms because they're built up by addition of 2 carbon units, and they tend to be unbranched. We classify them by melting points. So fats tend to be those that are solid at room temperature, and oils are those that are liquid at room temperature. We classify them by their saturation level, meaning how much hydrogen do they have. So if they have as many hydrogens as they possibly can, they're referred to as saturated. If they have double bonds, then they tend to be unsaturated. And we also classify them by the orientation of their double bonds. And so most natural fatty acids tend to be in the cis conformation. And you can see that all that goes together to help define some of the physical properties of these fatty acids. So up here, we've got stearic acid, no double bond. And so what you do is just get that staggered conformation of a hydrocarbon chain, and it's got a really high melting point. And the reason for that is it can pack together well with other hydrocarbon chains. So I got these pencils over here, so these can stack together. Got lots of surface area contact, and so it has a very high melting point. Like I said, natural fats, when they have a double bond, tend to be at the cis conformation, and so they put a kink in the backbone there. So oleic acid here has 1 double bond, and its melting point drops down to 13 degrees Celsius. Linoleic acid actually has 2 double bonds. Its melting point drops down to -5, and linolenic acid actually has 3 double bonds. And so in that case, it drops quite a bit down to -11. And so you can see that that conformation, especially around that cis double bond, plays a large role in the physical properties. And so I say for these that have lots of kinks in them, it's more like packing tennis balls where you've got very little surface area contact. And so not a lot of van der Waals interactions there. And so they tend to have very low melting points. So let's just look at some C18. These are the ones that we just looked at before. A little bit more about how we number and describe these. They're numbered starting with the carboxylic acid end, but they also have this other numbering that they refer to this end as omega. That is also used in mathematics to symbol but. just like that equal is equals, and that is therefore. In math, you may see this, so that is, but, okay. So omega carbon, that is the last carbon. You may have heard of alpha and omega, the beginning and the end. So omega is the end carbon right there. We'll see where that comes into play in a second. Most carbon, or most fatty acids in biochemistry have between 14 and 20 carbons. And again, they tend to have even units. I just read somewhere, and I can't find it. To be honest, I haven't looked that hard, but someone just discovered an odd numbered carbon or fatty acid, and they're actually starting to sell it this fall as a supplement, and it's supposed to have protective properties or be beneficial. So we'll make that the bonus. Find out what that fatty acid is. I'll give you a hint. It's got odd number of carbons in it. And it was some lady, and like I said, I can't remember where I read this story, but it was within the last week. That shows you how bad my memory is. But she set up a company and is now moving to sell it as a nutritional supplement. Anyway, I thought it was interesting that they found that in our bodies we do have odd carbon chains, and they may play an important role. So the bonus is to find that person and how many carbons is in that odd number of fatty acid chain. Okay, so like I said before on the previous one, we categorize them whether they're saturated or unsaturated or polyunsaturated if they have many unsaturations. Excuse me. So these all have 18 carbons, and you can see that we go from having one double bond. Again, that's in the cis configuration. And you can see that this drawing here isn't really indicative of the true structure. It almost looks like it's just linear. These pictures here are, oh, sorry, it froze. But the pictures on the previous slide are much more indicative of their structure. They actually have quite a big bend in them when you start to get multiple double bonds in there. Okay, so this is what it's going to show you is, yeah, once you get those multiple double bonds in there, you start to get kinks. So this diagram right here really isn't showing you the proper three-dimensional nature of these. So there's a whole list. Again, the study guide will direct you to those that you should know. And so you can, again, classify these by the number of carbons in there. It's easy for the saturated fatty acids because you just got 12, 14, 16, 18, 20, 22, 24 carbons in there. You can see that as we increase the number of carbons, the London dispersion forces or the surface area contact between them increases, and so the melting point goes up in a pretty regular fashion. Now, as you start to include double bonds, they're all cis, and you can see that you get this play. So here we've got 16 carbons and one double bond, and then we've got 18 carbons and one double bond. So it makes sense as we add carbons, it goes up. As we add double bonds, the melting point goes down. So you can kind of pick out some trends here. So these 18 carbons right here, as we increase the number of double bonds, the temperature goes down. In fact, these two here both have 3 double bonds. It's just the location of the double bonds. And what this is indicating here, so it's 16 carbons, 1 double bond, and the position here, this N-7, you can see that down here for unsaturated fatty acids, the quantity N-X indicates the position of the last double bond in the fatty acid, where N is the number of carbons, so that would be 16 minus 7. So that would be that the double bond appears at position 9. And you can see that in the name, 9-hexadecanoic acid. I don't know why they use this one here. Sometimes they also refer to it as delta 9, or these are omega-9 fatty acids. So a bunch of different ways to indicate where that double bond is in the fatty acid. So to me, instead of using this formula over here, it's easier to just use this number there. But yeah, this is the position of that double bond. Actually, I think I lied. Sorry, I think I made a mistake there when I talked about omega-9 fatty acids. Sorry, this is the position from the omega. This is the position from the carboxylic acid, and then to figure out the omega, you actually have to figure out from the end. So this double bond, sorry, my math was bad. This double bond appears at 1, and then you've got 5. So this is a omega-7 fatty acid, if I'm reading that right, which seems really weird. Let me continue on, and we'll catch up to that later. So common patterns in carbohydrates, they differ depending on the type of life that you have. So in eukaryotes, our fatty acids tend to be just straight chain, and more than half tend to be unsaturated. In bacteria, they actually have a lot more variety in their fatty acids. They are rarely polyunsaturated, meaning have multiple double bonds, but they do have branching, and they do have hydroxylation, and they can even include these cyclopropane rings. And our archaeal lipids are isoprenoid, which we'll need to talk about, and they're modified. And they actually have a lot of interesting properties for these bacteria, and we'll talk about that later. So something else about the double bonds is they tend not to be conjugated, meaning you have a double bond, single bond, double bond. They tend to be separated by at least 2 sigma bonds there. And so this is where I was getting tripped up. So the first double bond is commonly between C9 and C10. So what we do is we call that a delta 9 or a 9 double bond. So this is counting from the carboxylic acid end there. Now, another way is you count from the CH3 group and count in, and you figure out how far it is in. So you may have heard of omega-3 and omega-6 fatty acids. So this is counting from the opposite end of the fatty acid. This is counting from the carboxylic acid end, and this is counting from the terminal end, or the CH3 end. So 2 different ways to locate the position of the double bond there. And so in nutrition and popular literature, you'll hear, omega-3 and omega-6 fatty acids are healthy. But in chemistry, we tend to use kind of a systematic approach where we're counting from the highest priority functional group towards the end there. Anyway, just be aware that there's 2 different ways to indicate the location of the double bond, either starting from the carboxylic acid end or from the terminal methyl end. But also remember that these double bonds tend not to be in conjugation there in these fatty acids. So, like I talked about before, these fatty acids tend to stack together very well and have higher melting points the more carbons you have. They nestle together really well like Pringles, and so they've got a lot of surface area contact. And again, like I said, this is a fairly consistent trend. As you increase the number of carbons, the melting point goes up. As you put double bonds in here, the molecule tends to put a kink in there. And again, if I look between here and here, I've got 2 different trends going on. I've got double bonds in there, but I'm adding carbons. So this is constant, the double bond, but I'm adding carbon, so it makes sense that the melting point goes up. Now, in this next one, I've got the same number of carbons, but I've increased the number of double bonds and the melting point goes down. I've increased the number of double bonds and the melting point goes down. And now here I've changed two variables, and so that may be why it doesn't make sense, but I've increased the number of carbons and I've increased the number of double bonds. And you can also increase their position of double bonds. So it's hard to come up with the trends here if you're not keeping things constant. The take-home message is the more double bonds, the lower the melting point is, and that's due to this cis nature of the double bond and adding kinks, which prevents them from packing together fairly well. Now, like I said, those fatty acids, they do play a role, but really the biggest role, or where you find these fatty acids, is in triacylglycerols or triesters of glycerol. So if you take an alcohol and a carboxylic acid and you esterify them, you get an ester. So if you use this glycerol molecule here, you end up with what is more properly known as a fat or an oil. So I did mention that before when we're looking at fatty acids, but really when we talk about fats and oil, we're talking about triacylglycerols. The definition is still true. A fat tends to be solid at room temperature, and an oil is liquid at room temperature. And there is a connection between the fatty acid and the triacylglycerol. So here's an example down here at the bottom with all the carbons drawn out, and you can see that this has a saturated chain. This one has one double bond, and then this one has two double bonds. Now they do draw it out like this, but again, I can't stress enough that with these double bonds in here, there are kinks in that structure. And so that actually plays a huge role in the properties of these double bonds, or sorry, of these fats. Sorry, I just lost my screen. So, as I mentioned before, these fatty acids are typically found as triacylglycerols, and they are used to store energy in our body. So we can actually get 9 kcals per gram out of these fats. And to process them, like I mentioned, we store them as triacylglycerols, but we do hydrolyze those esters, release the fatty acids, and then we metabolize them. So these fatty acids aren't very highly oxidized. They just tend to be oxidized at the carboxylic acid and or where the double bonds are, where if you look at carbohydrates, they have a lot of oxygen on there. So they're already what we could say, halfway oxidized. So when we continue processing carbohydrates, we get less energy out of them than we do out of the fats. And the reason for that is fats are less oxidized, so we can get more energy out of them as we oxidize them. So as I mentioned, these triacylglycerols are stored. They're stored in cells called adipose cells. And adipose cells are really mainly just fat. So they've got a little bit of other machinery and nucleus in there, but they're really just globules of fat. How I liken this is if you've ever had kind of a fatty soup, an Italian sausage soup, you'll see those little fat globules that float to the top. And if you get bored and you don't like the soup, then you start connecting those globules and you see how big of a oil streak you can get on top. Or if you actually go and put it in the fridge, those will freeze. And anyway, kind of the same type of idea. You've got these essentially fat globules inside of membranes there. And so when we need to release the energy, we go in, hydrolyze them, release the fatty acids, metabolize them, release CO2, water, and a large amount of energy. So a little side note, so you may have heard when you were young that the camel's hump stores water. Well, the camel's hump really stores fat, but in breaking down fat, one of the byproducts is actually water. So it's not entirely false that the camel's hump stores water. It stores it in the form of fat, and when it's metabolized, it produces water. I don't know if the camels actually get much of that. They do have ways to recover the water from their breath. So I'm not sure really how much the water from the metabolism of fat benefits them. But anyway, I just remember people tell me, no, that was a lie. And I go back and say, no, it's not really a lie. You now have greater knowledge. So you can say, yes, the camel does store water in its back, just in the form of a fat. Okay, so they're used as energy storage. So some other interesting things is just where do we find saturated fats and unsaturated fats? And this slide just more for those that are interested in nutrition. So to me, the saturated fats are always the ones that taste good. You look up here and you're like, yummy. You look down here and you're like, ugh, that looks healthy. So again, if you have these saturated fats in there, then you tend to be a solid at room temperature. So you tend to find these type of fats in animal products, such as dairy products and meats. If you start putting double bonds in there, these don't pack together very well, and you start to find these in plant sources, and those are referred to as oils. And again, you can kind of see the distribution of saturated and polyunsaturated. So what's interesting is coconut oil is thought to be healthy, even though it has a high amount of saturated fat. One of the reasons that is thought is because that coconut oil has a short chain saturated fatty acid. So we really are still trying to figure out nutrition. And, one time butter was bad and margarine good. Now butter's good, margarine's bad, eggs are good, eggs are bad, eggs are good. Saturated fats are bad. Now all of a sudden saturated fats are good as long as they're short. So still a lot to figure out about what's good for us and what's bad. One of the things that we know that is bad for us is trans fats. Now, these aren't naturally found in large abundance. There are some cases where we do have trans fats, but they're not really a high abundance fat. Now, with that trans double bond, they look very similar to just a saturated fat. And in fact, they pack together just like saturated fats. And what happens is when you take a unsaturated fat here, these tend to absorb oxygen and can go rancid, and they tend to be oils. And even though they're healthier, they're a little bit harder to deal with. And like I said, they can go rancid, or they're hard to put on your bread. So what they do is they partially hydrogenate. They do a reaction with H2 and some type of catalyst, typically nickel. And when they add the hydrogen across there, you get some rearrangements, and so you actually produce a trans double bond. And our body's not set up to really process trans double bonds, and so it's thought that these are detrimental to health. These were found in highly processed food, like the interior of Oreos and margarine and Crisco. And now things claim that they have 0 grams trans fat. Well, you got to be careful when something says 0 grams trans fat. That means that they could contain 0.5 grams trans fat, but what they use is one significant digit and call that 0 grams trans fat. And I believe if they say no trans fat in there, then it doesn't have trans fat. But anytime you see this 0 grams, beware, because it likely does contain trans fat. It just contains less than 0 grams. And I think the cutoff is 0.5. So you can have 0.5 grams trans fat and still say that your product has 0 grams trans fat and I believe that's per serving as well. Anyway, trans fats are bad for us. So let's continue on. So we talked about triacylglycerols. Triacylglycerols are simply where we have this glycerol molecule, and that's shown in black here. And we have a fatty acid appended to each one of these. Well, in this case here, we don't have a fatty acid. Off of this position, we actually have a phosphate. And so these are known as glycerophospholipids. We're going to have fatty acids here as ester linkages. And then this one here, we actually have a phosphate. And then off to this group here, we actually have various groups. And so these are known as glycerophospholipids. R1 up here generally is a saturated fat between C16 and C18. R2 is often an unsaturated fat between C16 and C20. And X is usually a polar alcohol. And so you can see that you could have ethanolamine, which is, you'd have an oxygen here, and then at physiological pH, your amine would be protonated. Choline, which is always protonated because it is a quaternary amine. It's got 3 methyls around there. You could have appended the serine amino acid. So the side chain is actually attached to that phosphate. Myo-inositol, so an interesting sugar that doesn't have an oxygen in the ring, but you are using one of the oxygens off the ring. You can have just glycerol. So you'd have a glycerol backbone, and then you'd have another glycerol molecule out here. And those can also form some interesting compounds, because then you could get another triacylglycerol to join here. I believe cardiolipin, oh, there it is right there, is actually kind of a combination of kind of a dimerization of 2 fatty acids through this glycerol. So you got this fatty acid here, you've got this phosphate. This phosphate's connected to this glycerol here, and then you have another fatty acid portion. So yeah, cardiolipin is kind of a dimer of these glycerophospholipids. So all sorts of groups that you can put off of these phospholipids, and we'll talk about their functions a little bit later. So I just thought it's easier to see some of those structures. So you can see phosphatidylethanolamine, phosphatidylcholine, phosphatidylserine, and phosphatidylinositol. And so again, this is glycerol, and then off the glycerol, you would have 2 fatty acid chains there. So this is just showing the structure of the head groups. So these are known as phospholipids. And so here is a full structure of a phospholipid and a computer modeling. So again, you tend to have in position 1 just a saturated chain, and you can see how that just is pretty straight. In position 2, you have cis double bonds, and notice how that includes a kink in it. Then you got the phosphate. And again, this kind of a computer model, so things are kind of wrapped around itself, but there's a phosphorus atom and the 3 oxygens around it. And then you've got the head group on there. And so we're going to use these to make bilayers in there. So what's interesting is when you throw these in water, you've got this polar head group and these nonpolar tails. These nonpolar tails want to get away from the water. Or another way of looking at it is the water would be very organized around these hydrophobic tails. And so to maximize entropy, you want to minimize the contact with water. So we're going to see the hydrophobic effect again. Before we do that, let's just talk about an example of a glycerophospholipid that actually is important. Dipalmityl phosphatidylcholine. So palmitate is a 16 carbon chain saturated. So you've got 2 of these chains, and then you've got a phosphatidylcholine. So let me just go back and show you what the phosphatidylcholine is. So you got this phosphatidylcholine, and so actually let me go back here. So you'd have this phosphatidylcholine, and then you got these palmityl groups, just 16 carbon saturated chains there. And this is an important molecule. It's a lung surfactant. Surfactants are surface active agents. And so as the air escapes out of your alveoli in your lungs, you don't want the lungs to collapse and stick together. That takes a lot of energy to expand that airspace. So really what you want is for the air sac to contract somewhat and the air goes out, but you want the air sac to remain rigid. You'll still have some air in there. And like I said, you don't want to empty it out completely like a balloon. There's lots of issues. The membranes can stick together, and anyway, it's just harder to inflate. So what they do is they have this dipalmitylphosphatidylcholine that actually is at the interface of the alveoli and the air. So in fact, the polar head groups are pointed inward and the tails are pointed out towards the air there. So there is a thin layer of water and you get these nonpolar head groups, and they pack together fairly tightly here. And so as the air escapes, those glycerol phospholipids pack together and prevent the alveoli from collapsing. And so you can kind of see that as you got the fluid and then you've got these bilayers there to prevent it from collapsing. So as I expand, as you breathe air in, the space between the surface or surfactants would actually increase. And then as you let the air out, they kind of come together and compress, and they just don't let the alveoli from collapsing. Okay, so interesting uses there. We'll get back to membranes. We've got a whole section on that, but something else we need to talk about is phospholipases. These are enzymes that actually hydrolyze glycerol phospholipids. When I was an undergraduate, I was just fascinated with these. The reason is because I hate bees, and when bees inject you with their venom, they actually inject you with a phospholipase. And so they categorize these phospholipases by the type of bond that they cleave. And so you have phospholipase A1, phospholipase A2 actually cleaves that bond. Phospholipase C cleaves the bond before the phosphorus, and phospholipase D cleaves after. And so snake venom and bee venom contains a lot of phospholipase A2. These are the best or most well studied. And so what they do is they actually release the fatty acid off that middle position there. And they end up with what are known as lysophospholipids. Now what's interesting are these start acting like surfactants. And the reason for that is you no longer have this OH there. So what you do is you get kind of this long fatty acid chain. You got this glycerol that has an OH there. And then you've got this polar head group here. And so it actually just acts more like a detergent. You don't have the two chains there. And so once you cleave off that middle fatty acid, this ends up acting like a detergent and causes your cells to kind of just wash away and lyse. And so all of a sudden you get lots of your cells breaking around that site of the injection of the bee or snake venom. And anyway, that as you can imagine, causes problems. So snake venom, it catalyzes the hydrolysis of the phospholipids. Like I said, there's a couple of different places that these phospholipases can break. But like I said, the diamondbacks and the snakes tend to release at the phospholipase A2 region. And there's something else that's interesting when you release this. There's often a unsaturated fatty acid here. And one of those unsaturated fatty acids that can be found here is arachidonic acid that leads to my favorite molecules, the prostaglandins. So we'll talk about that in just a second. But anyway, these phospholipases, here is a cobra phospholipase. And what's interesting is that phospholipase has to kind of pull the lipid out of the membrane, which we'll talk about, and get in there. So it's got these hydrophobic residues that kind of interact with these hydrophobic tells and pulls it out, and then it can do the cleaving there. Okay, now there is a lot of variety in lipids. Like I said, just the structural definition gives you a lot of variety, but in these, I guess, phospholipids, there's some variety here. So if you look at this molecule off to the right called the plasmalogen, it has a different functional group. So it's still got the glycerol backbone here. It's still got the acyl group, meaning a fatty acid. It still has the phosphate, but if you notice, it has an ether linkage here. And that ether linkage is actually to a double bond. So it's actually a vinyl ether. And vinyl ethers are easily oxidized. The full importance of this isn't known, but plasmalogens are thought to help us deal with oxidative stress. And so what happens is this site gets oxidized instead of other places, and then it gets regenerated. So again, even within glycerol phospholipids, there is some variety. Like I said, you get the variety of the groups attached to each oxygen, you get the variety at the phosphate head group, and now we actually get variety in the linkage to the glycerol backbone. So those are glycerol phospholipids, glycerol because they come from glycerol. We also have a new type that we need to talk about, and these are called the sphingolipids, and they are derived from sphingosine. Now, sphingosine, if you squint hard enough, you may think that, oh, this is kind of like a glycerol molecule there. We've got an oxygen, nucleophile oxygen, but notice that we actually have a covalent bond here. So this is actually a single molecule called sphingosine. These are all carbon-carbon bonds along there. And so this sphingosine can actually set up very similar to the glycerol phospholipids, just different bonding patterns. And so some of those different bonding patterns, like I said, the whole backbone is a single molecule there. And then off of the nitrogen here, instead of forming an ester, you can form an amide. And when you put a fatty acid off there, it's now referred to as a ceramide. So ceramides actually have the sphingosine and a fatty acid residue. And guess what? We're going to include some complexity here, and we're going to attach all types of groups. So we're going to be throwing out a lot of names, but you just need to understand that sphingosine is kind of the base molecule. And so these are called sphingolipids. They were mysteriously, they weren't quite sure their function, so they got the name Sphingo from the Sphinx in Egypt, which is just kind of its recognition that these are mysterious molecules, or at least they were. So we got sphingosine, which is the base, ceramide when we put a fatty acid off there, and then at this yellow position is going to give rise to a bunch of different molecules. So here's a slide. I didn't finish putting it together, but essentially you've got a fatty acid, you got sphingosine, you've got the phosphate head group that we're going to put on there, and then we're going to put different groups. So this yellow phosphate is what we would attach at this yellow spot right there. And then we're going to have all sorts of groups hanging off that phosphate, just like we saw in the glycerol phospholipids. Okay, so our first example are the sphingomyelins. These are the most common sphingolipids, make up 10 to 20%. The head group is, again, a phosphate ester. So just like we saw in the glycerol phospholipids, let me get my color coding. So here is the yellow, and we're going to put a phosphate on there. So actually, if you overlaid this with the glycerol phospholipid, you'd see that it's very similar in general structure. And then we add a choline group, which we did to the glycerol phospholipids as well. Now, these are sphingomyelins. These are found in the myelin sheath around electrically insulating cells. So many nerve cell axons are rich. So this is a picture of the myelin sheath, and we'll talk more about that later. We actually have many layers there, and that acts as an insulator. So these cells can actually conduct electricity. So if you look at the overall shape of this, it looks a lot like a glycerol phospholipid. Okay, so you can also attach sugars directly to that alcohol. Now, you don't have the phosphates like we saw in the glycerol phospholipids, but you can attach sugars there, and these are called cerebrosides. So they're important components of nerve, muscle, and skin membranes, and so they can hydrogen bond with polar head groups. So they have a big impact on membrane properties. Now, they're not charged because they don't contain the phosphate. We're lacking the phosphate group here. We're connecting the sugar directly to essentially the ceramide there. So the ceramide, because, let me do the coloring again. So this is the sphingosine right here. The ceramide is when we have that group right there. And then now the cerebroside, because we're adding a carbohydrate on there. So those are cerebrosides. And you can expand out and put multiple carbohydrates on there, and those are called gangliosides. And these are often found in abundance in our brain. So these oligosaccharides, like we talked about in the last chapter, they extend out of the membrane and they act as receptors for glycoprotein hormones. So proteins will come along and recognize these oligosaccharides sticking out of the membrane. And in fact, some bacterial protein toxins, the cholera toxins, actually recognize this. And that's a bad thing because the cholera toxin will recognize the cells and then wreak havoc. Okay, so most cells continually degrade and replace their membrane lipids. So we actually regulate the composition and the placement of those type of lipids. And like I said, we got those phospholipases that can cleave off groups, and then we can append new groups on there. And the gangliosides are degraded by a series of enzymatic cleavages. And there's actually a lot of diseases that are based on improper handling and metabolism of the gangliosides. And these are referred to as lysosomal storage disorders. So actually quite a few different examples of those. And here is just a model of a cerebroside. So you can see that it's got a bunch of carbohydrates up here. And then again, you've got your sphingosine, the ceramide right there, or the fatty acid chain in there. So just a three-dimensional representation. So we'll come back to these membranes, or sorry, these lipids when we talk about membranes, but we just got a couple more different lipids to talk about, and one is steroids. So you should know the steroid core. So it's a 6-6 offset 6 5-membered ring. So you should be able to draw this general structure, not necessarily with those methyl groups and that R group off there, but you should be able to draw the A, B, C, and D ring and recognize that that is the steroid core. So plants, yeast, fungi synthesize and use steroids. Prokaryotes don't use steroids as much, and in animals, we use it a lot. In fact, membranes, again, that we'll talk about soon, are 30 to 40% made up of cholesterol. And cholesterol ends up having kind of a flat, rigid structure, and so it's used to control membrane fluidity, which again, when we get into membranes, we'll talk a lot more about. So you can see cholesterol down here in its general structure, but again, notice that 6-6-6 5-membered ring form. And so the steroid hormones, these are molecules that modulate biological processes. They're classified into 3 different groups. The glucocorticoids. One example is cortisol that affects carbohydrate metabolism. So you can kind of pick that up from the name, the glucocorticoids. You got the mineralocorticoids, and so these regulate the excretion of salt and water in the kidneys. And that kind of makes sense, the mineral. And then you got the gonadocorticoids, and these affect the sexual developments. And that kind of makes sense from the gonads. And so in the gonadocorticoids, you've got the androgens, and these tend to be the male sex hormones synthesized in the testes and regulate the development and maintenance of male characteristics such as hair and deep voices. And then you get the estrogens and progesterones. These tend to be synthesized in the ovaries and regulate the development and maintenance of the female characteristics and tightly involved in pregnancy and menstruation. Okay, so in sterols, you can actually use them to make vitamins. And I just had a technical glitch. So I've just got a few more slides, but I'm going to end the lecture here. So I just need to talk a little bit about the vitamins that come from steroids. We need to talk about isoprenoids. And then I need to talk about the eicosanoids, or the prostaglandins, which are my favorite molecules. And I really don't want to rush that. So I'll have that at the start of the next lecture. There's not much left. So like I said, we just need to talk about steroids. And then what we're going to do is take these lipids and make them functional. And what I mean by that is we're going to put them together to make cell membranes, which, if you remember way back when we were talking about the origin of life, we needed to find a way to compartmentalize these reactions. They just really couldn't happen in solution. We need to find a way to compartmentalize them so we can concentrate and control and regulate them. So cholesterol, these glycerol phospholipids, these sphingolipids, they're all going to go into constructing cell membranes. So we'll pick up, like I said, talking a little bit more about steroids and some isoprenoids. I'll talk about prostaglandins. There's not much in the book on it, but like I said, they're my favorite molecules. And then we'll jump right into membranes in the next lecture.